How's it going? Good. I'm all right. Okay, good morning. Welcome to the last cell lecture. Yay. I'm as happy as you are. Okay, so most of the details of the poster session, all that's been covered in Piazza, so if you need instructions, be sure to go look at that, because we really need to stick to the lecture. Probably will not have time for a break, but I have to get to a point where I can just say we're going to cut it right here. So those of you that came to class today are going to know what's going to be covered, and those of you that did not will not know. Okay. One thing I do want to point out for Thursday's poster session is be here early. This room is actually empty before our session. It begins at 9:30. I'm starting the poster session at 9:30. So that means if you're a presenter, you need to be here no later than 9:20. If you're a viewer, you need to be here no later than 9:20, okay? Because you have to go, and there'll be easels set up all around this room, and so you have to go find your easel where you're supposed to be, whether you're a presenter or whether you're not, okay? So if you haven't looked at those rules and what you're supposed to do, look at them on Piazza and the files that I posted. And if you have questions, post it on Piazza so everybody can see that, and we'll help answer those questions, okay? Also, there is one more evaluation that you have to do for your group members. It's the final ranking. The people that present first on Thursday, your rankings are going to be due Friday at midnight, okay, or Friday at 11:30. So when you get that, finish the poster session, that's when you can start ranking your group members. But you need to finish it right away, okay? There's no reason to drag that out. It should take you a few seconds to do that. And we'll do it the same way we did last time. Was it an assignment, the midterm ranking? Yeah, so I'll post it. It's not up yet. And then, so the presenters, you're going to do your rankings first. So the Cures 1 will do theirs first. Next Tuesday, you can't rank your group members until everybody's presented. Okay, so the second group will, when you're presenters, then your final ranking of your group will be due Wednesday at some point, okay? All right, any other quick questions before we go on? Okay, so what we have to do today is finish up briefly the cytoskeleton. We've talked mostly about microtubules. We're going to finish a little bit with that. We're going to go into a little bit about microfilaments, and then very quickly go into the cell cycle. So a couple of different topics today, and we'll try not to slow me down if I'm going too fast, but all of the information is actually on those outlines, so they're pretty detailed, okay? The thing that we didn't talk about with microtubules is what gives them their functional diversity, and that is the proteins that bind to them. They're called MAPs, microtubule-associated proteins. And there's lots of different kinds of MAPs, but we're going to focus on those that stabilize the microtubule, okay? And so a typical one in neurons, that's where you're going to really need a very stable microtubule highway. What do you think is going to move down that highway in an axon that's in a neuron? Pardon me? Yeah, but they're going to be inside of vesicles. Okay, so you're going to have a motor protein, and we're going to look at how that works, carrying the vesicle, walking along a microtubule. What if that microtubule is unstable? You can't neurotransmit anything, okay? So you need to have these proteins that stabilize. And there's two varieties. In neurons, there's one called MAP2, and there's one that's called Tau, okay? And both of these have the same type of setup. There's a microtubule binding domain, okay? And then there is a projection domain. The size of the projection domain determines the spacing between the microtubules. So this is a cross-section through an axon, and you're looking face on to the microtubules coming in and out of the plane of the picture. And the reason the microtubules have a wider spacing in this shot as opposed to that neuron is because of what binding protein is there. So MAP2 is in this neuron, Tau is in this neuron, and the projection domain on Tau is much shorter. So the packing of the microtubules is closer together. Why do you want to kind of space these out a little bit if you're in a neuron? Remember, this is the highway, but stuff has to walk up and down this tubule, right? So that's going to be, the only thing that you have to have that space there so that the motor proteins can move up and down and carry stuff. Everything from mitochondria to neurotransmitter laden secretory vesicles. Okay, so the spacing is determined by the projection domain. Now, the reason this projection, the microtubule binding domain sticks to the microtubule is that it has at least 4 basic residues that are positively charged. Okay? And the reason that's important, that's not just a picky detail. If you phosphorylate this part of the map, then what will happen is you neutralize those positive charges and the MAP2 will unbind. Okay? That's going to allow the microtubule to fall apart. And we're going to need to do that to microtubules when we push the cell into mitosis. And there's a reason for that. But here's the key thing. If you phosphorylate this, it's going to unbind from the microtubule. The microtubule will become unstable. Now, in Alzheimer's, what happens is if this thing gets hyperphosphorylated, it unbinds and it can't rebind. In fact, what it does is it binds to another tau and forms what are called neurofibrillary tangles. And that's one of the hallmarks of Alzheimer's, the presence of those tangles that are made up of paired tau filaments stuck together instead of to the microtubule. And that causes the neuron to swell. It screws up the neuron's ability to transmit information, and you get Alzheimer's. Okay? It's not probably the cause, but it's coincident with what's going on in a neuron that's undergoing Alzheimer-like changes. Okay? So it's important stuff. Now, the other real important map is motor proteins. That is, those motor proteins are going to move stuff in different directions along the microtubule. Okay, so here's the negative end of a microtubule. This is the positive end. If this is not part of a cilia or flagella, where would this be located, the negative end? It would be projecting. Pardon me?
The microtubule assembly center?
MTOC, microtubule organizing center. And what do you call that in most cells? The centriole. Okay, so that's going to be oriented by the nucleus. This is going to project to the plasma membrane. So if you want to send vesicles to the plasma membrane, say like GLUT4 secretory vesicles, okay, you load them up here, you bind it to this machine, which is called kinesin. Kinesin is a microtubule motor protein that walks to the positive end of microtubules. What if you want to traffic stuff to the negative end? Okay, let's say you have a protein that is a transcription factor, and you want to take it right to the nucleus. And so it binds to a different motor protein called dynin. Dynin walks to the negative end. So you choose the motor protein depending on which direction you want to go. Okay, is that clear? All right. And we're gonna detail, this is going to be our poster child for how this thing operates. We'll do it after we've already covered microfilaments here in just a few moments. Okay, so that really concludes our information about microtubules, and we will come back to the motor proteins. And so now what we're going to talk about is microfilaments. And a lot of you know that microfilaments are involved in muscle contraction, smooth muscle and skeletal muscle contraction. Okay, the myofibrils contain just series and series of microfilaments interspersed with motor proteins called myosin. That was one of the first proteins we talked about, was myosin 2. Okay, so skeletal muscle contraction is due to interactions between microfilaments and motor proteins. Right, so that's obviously important. movements of cells along substrates inside the body. It's the ability of cells to crawl is determined by microfilaments. Polymerizing at the front end, they polymerize, that causes the cell to protrude. You depolymerize at the back end, so there's this flow of actin to the front, creating projections that are made out of microfilaments. So you poke out the front, you depolymerize in the back. And so this flow of actin actually pushes the cell along. Okay? So cell motility. Are microtubules involved in cell motility? Very special cells. How does a sperm move? Yeah, so a flagella, the reason the sperm moves is because of microtubules moving, okay? So both microtubules and microfilaments are involved in cell motility. But if the cell's going to crawl, it's going to be microfilaments that are going to do the job, okay? One of the most important things we'll talk today is another function of microfilaments. And microfilaments form a contractile ring. This is actually almost identical to what happens in skeletal muscle. So there's a belt of actin filaments, and then there's myosin motor proteins that are walking along those filaments that squeeze this ring just like a purse string. And that's what actually causes cytokinesis. So microfilaments are important for the division of the cytoplasm. See, there's two types of cell division going on during mitosis. There's nuclear division, that's called mitosis proper, and then cytokinesis is splitting the cytoplasm into, that's called cytokinesis. But microfilaments drive that squishing of the cytoplasm into two different pieces. Okay, good. So that's some broad functions of microfilaments. Now let's remind ourselves of how the fundamental unit of a microfilament is different from a microtubule. Okay, so again, it's really important for the final exam purposes you be able to compare and contrast these different filament types. Okay, so again, this one binds a nucleotide just like microtubules. Hhowever, this is a monomer. It's called globular actin, G-actin. Okay, it's got four sort of alpha helical domains, but there's a little notch in here. Okay, and in that notch is where an ATP can swim up here and bind. So this is the nucleotide binding cleft. And that cleft always faces what we call the minus end. The positive end is on the opposite side of G-actin. And the reason that's important is you actually build the filament from the minus end up to the plus end. Okay, so at the minus end, what you're seeing is the cleft for ATP right there. And at the top of this is the top end of that last actin, G-actin. Okay? And when it's all aligned in a filament, which is really tiny, it's the smallest of the filaments in terms of diameter, what happens is this thing can undergo that same type of dynamic instability that microtubules go. Okay? Because once ATP is bound to G-actin, or in this case, what's really interesting is that ADP bound to G-actin can also bind. Okay, so that's different. What happens in microtubules? You have an alpha beta tubulin heterodimer. They both have to be bound to GTP in order for that to bind. That's not the case here. Okay, so why the difference? What happens is at the positive end, an ATP-bound G-actin will bind faster than an ADP-bound G-actin. Okay? At the negative end, they bind at the same rates. Okay? An ADP is just as good as an ATP-bound G-actin. So what happens is that means that the addition of new actin G-actins is faster at the positive end than it is at the negative end. So it ends up being just like a microtubule, but for slightly different reasons. Okay? And once those, if you have an ATP G-actin, once it gets into the tubule, it begins to hydrolyze the ATP to ADP. If it joins this filament already in the form of ADP, it doesn't need to do that, right? It's already been hydrolyzed. But the same rule applies. If there is a cap of ATP-bound G-actin, this thing is very stable. But the minute that cap is hydrolyzed to ADP, this thing starts to be very unstable. Okay? All right, the nice thing about it is usually you need very stable microfilaments, and there are proteins that help it stabilize, right? So that's the basic biology. And we can actually tell, you can't tell by looking at actin filament which direction it's running, but you can if you use a little helper. Okay, so this is myosin 2. This is a motor protein. Okay, this is the actin binding domain that's going to bind to ATP and split it to move this motor protein along the microfilament. Okay, so what if in the laboratory we can actually chop this up into little pieces until we get just the actin binding domain, which is this part right here. Okay? And what happens is if you mix that with one of these guys, those little head groups will stick to the actin filament because that's the active binding domain. Okay? And what it looks like is it forms a filament that looks this way. It looks like it has, it's hard to see, but if you look at it up close in your PowerPoint that you get, those little arrowheads. Okay, they're shaped like little arrowheads, and I drew a big one there just to make sure you understood what I meant, okay? And the arrowhead is telling you which way the filament is oriented, okay? So here's the rule. That arrowhead, if it's the pointed end of the arrow, the direction that the arrow is going to fly, that's aimed at the minus end, okay? We call this end the barbed end, and the barbed end is oriented to the plus end. Okay? So you can always tell where the plus end is versus the minus end by which way the arrow is pointing. Okay? And you make these arrows by loading in these subfragments of myosin onto an active filament, and we can tell which way it's going. Okay? But remember, the plus end is going to be polymerizing fastest, and usually the minus end is the slowest. It's always the slowest. Okay, questions so far on the basic? And this orientation is important because the way it's pointing determines how it's going to protrude out of the cell. Usually, if you want to form protrusions into the plasma membrane, that's usually the positive end because you're going to polymerize here faster, so you use that end to point towards the plasma membrane. Okay. But the nice thing about microfilaments is they don't have to be nucleated by a microtubule organizing center of any kind. There is no such thing as a microfilament organizing center. They can polymerize at any point in the cell. That's what's really cool about them. They're more versatile in where they begin and where they end. So that's a big difference between them. Okay. All right. Now, just like for microtubules, what gives microfilaments their diversity is the binding partners, like myosin 2 is a binding partner. Okay, so they're MFAPs, microfilament associated proteins. Okay? And so we're going to look through, very quickly, we're going to go through several different kinds of those proteins because they control the function of the microfilament system. Okay? Now, this one's called ProFillin, and it tells you exactly what it does. It binds to an actin, a G-actin, okay, loaded with some nucleotide, either ATP, typically ATP. And which side is it binding to? Which side of the G-actin monomer is it binding to? To the plus side, because that's the clef, the nucleotide binding clef, so this must be the positive end. So ProFillin has that name because it stimulates the addition of G-actin to the plus end, preferentially. So it helps load them onto the fiber, and it causes proliferation of the length of the microfilament. So this is something if you want to build a microtubule. Okay. Now, what if you want to slow it down? We have to go to a different picture, and so that's shown in one of these little, there we go. Okay, so this is, let me get this to be a little bit bigger. Sorry. All right. I don't know if there's any way to do it. Okay, I guess you can't get any bigger. Okay, so here's our friend ProFillin on this side. It's adding G-actin to the filament. Thymosin, it does just the opposite. It binds it and keeps it. It keeps it away from the filament. So we call that sequestering. So thymosin sequesters G-actin from binding. So this is a negative regulator of polymerization. ProFillin is a positive regulator of polymerization. So that's its function. Okay, so it's pretty straightforward. Now, do we have, let's go back to the PowerPoint. Okay, how do we nucleate? If there's no such thing as a microfilament organizing center, how do you nucleate microfilaments? Two different proteins are involved, and there's probably others, but here's the classic one. A protein called the ARP complex. It's actually made of two different kinds of proteins. It's often you will see ARP-23, ARP/23, because there's two different proteins. This ARP-23 complex is a nucleator of microfilaments, but it's got really special properties. First of all, it binds to the negative end, and it caps it. So it stabilizes the negative end, and then it nucleates the addition of different G monomers being added to the filament. Okay, so that's the first thing. It nucleates and it caps. The second thing, though, that's really important about ARP-23 is that it causes branching. So it can move up here, and a different ARP-23 will bind to a microfilament that's already there. Okay? But it always binds at a 70-degree angle and nucleates a new filament. What are we doing here? We're creating a network. So there's really two forms of actin once it's polymerized into a net-like group of filaments called a network, and that's exactly what it looks like. It looks like a net, okay? Or you can bundle it together. But to create bundles, we need a different sort of protein. We need a protein that's called formin, okay? And formin is, again, a nucleating protein, but it works very differently. Number one, it always associates with the plus end. So as this thing moves up, what it's doing is formin is stacking those G-actins inside of these two little ring structures. And so it just sort of wiggles back and forth. And as it moves up the filament, you elongate the microfilament length. So it works on the opposite end. It works on the positive end, not the negative end. And it doesn't cap it. It just moves with the filament as it grows. Okay? So that's a big difference between ARP-23 and this one. So this is going to work at the positive end instead of the negative end. The other big thing it does is if you want to form bundles, that's when you use formin. So we go back to this structure, and this is a table in your book. Let's back up here. See if it shows that. Oh, there it is. Okay, so what's happening, if you want to form a network, the rule is use ARP-23 to nucleate your microfilaments. If you want to form a bundle, use formin to build parallel bundles together. But you still need something to hold them together, and we're going to get to that protein that holds that bundle together. So formin builds bundles, ARP-23 builds networks. Okay? And so if you wanted to build a projection like a microvillus at the top of an intestinal cell, which of these nucleating agents would you use? If you wanted to create the little blips on Bart Simpson's head, formin. Because you need a tubular projection, not this thing that's spread out all over the place. Okay. Good. All right. And there's a couple other interesting things about formin is that it can cooperate with ProFillin and work together to get this thing to grow really fast. Okay. How does it do that? Well, here's formin up here, and there's like 8 or 12 different isoforms of formin, and some of them have whiskers, okay? And so these whiskers are hanging out here, and the whisker domain has binding sites for ProFillin. And so what happens is ProFillin loads onto the formin whisker, and the whisker goes like this into its head and puts the G-actin filament monomers on top of itself, and the thing wiggles up and forms a growing tubule or basic filament of G-actin, forming F-actin. Okay. So the whisker domain binds ProFillin. Those two are going to cooperate together. Okay. All right, questions so far. So that's the nucleation story for microfilaments. And so ProFillin, of course, helps in this polymerization. If we want to slow this down, we need to call upon thymosin, okay? Good. That's how simple it is. All right, now, so again, depending on what you're building, you may want to make two different kinds of bundles. You may want to make a bundle like this that's very close together. And how do you do that? Well, you use formin to create the microfilament strands, okay? But then you use a really short crosslinker. So this is about the crosslinker family. Short crosslinkers make tight bundles, okay? Loose bundles, you use a longer crosslinker called alpha-actinin, okay? And so where do you find these two things? You're going to find this in this structure here. That's this is one of Bart Simpson's little head things, okay, sticking out of his head. All right, this is a microvillus, and it has been created by formin, linked together with fembrin, and another one, which is aptly named, called villin, not because it's bad, but because it makes a microvillus. Okay? So short crosslinker makes a nice tight bundle, and that's what you need to make Bart Simpson's hair stand up. All right? Now, what do you do with this one? This is what you find in skeletal muscle. Okay? What happens is you need to space out the actin filaments wide enough to slide the myosin right inside here. So if you don't have enough spacing, you can't put the contractile machine in between the two different fibers. And if you can't do that, you don't have a skeletal muscle. You don't have a contractile unit. Because the myosins are going to reach up and bind to this microfilament and walk along it, and that's what causes muscle contraction. So this spacing is really important, just like it is for microtubules. So loose bundle, alpha-actinin, tight bundle, fembrin, and villin. Okay, questions so far? So we're starting to just build all kinds of stuff this way. All right. Now, what about even longer crosslinkers? Here's a really good medically important crosslinker, and this is dystrophin. And as you might have guessed, if this is mutated, you get muscular dystrophy. So dystrophin links the actin filament and cross-links actin filaments to the plasma membrane. So if you have an individual with, say, Duchenne's muscular dystrophy, and there's very different kinds of muscular dystrophy, and what happens is this thing is mutated somewhere, and so the actin filament is not properly anchored to the plasma membrane to give it stability. What's going to happen to that person's muscle cell when it contracts? It's going to rip. It's actually going to rip the plasma membrane because this thing is not properly attached to the membrane, and so it creates all kinds of damage and inflammation, and eventually the muscle cell dies. That's why you get muscular atrophy in the dystrophy. Okay? But that's the protein. That is a crosslinker, big, long, floppy crosslinker. Okay. One other one that we've looked at before, and that is in red blood cells, you have that sort of crisscross pattern. That is called spectrin. That's a real floppy actin crosslinker. So if you want a loose network of, you don't want a bundle, but you want a network, you can cross-link it with either dystrophin or spectrin. Okay? So those are the two floppy cross-linkers. And what would you nucleate that network with? ARC23. You wouldn't use form, would you? Okay, so certain things go together, others do not. All right, then this is one of the next to last pairings. What is this protein? This is tropomyosin. You find this in every single skeletal muscle cell. And its job is it's got two jobs. It stabilizes the structure of the actin filament shown here in red, or orange red, okay? And then this blue thing, which is tropomyosin, is called a side binder, okay? Side binder. And what it does is it controls access of the microfilament to the myosin head group, which you can't see. The myosin head group would be right here, and it reaches up to grab the microfilament, but if this is in the way, you get no muscle contraction, because it has nowhere to grab onto the microfilament, right? So this is actually controlling muscle contraction. And we don't have time to go into the details, but your book, if you're really interested in muscle contraction, the details are there. Many of you probably have already had that. But this is the mechanics of how that side binder is controlling the stability and the access of myosin 2 to the actin microfilament. Side binder. Okay. All right. Now we get to, so any questions about those things that we've talked about so far? You're really building a toolbox that you can put together in almost any situation. Make a cell crawl. You can make a network. You can make bundles. You can make projections, and control the cell function in that way. Okay, so molecular motors. This is always an interesting topic. This is a machine that converts the chemical energy of ATP hydrolysis into mechanical energy. It's really cool. So we got to get some kind of motion out of our ATP splitting. And so our poster child is going to be kinesin. We could use myosin 2. We don't have time to do that this year. We'll focus on kinesin, which is going to walk along a microtubule, and it will carry a cargo. Okay. Now, so there's all different kinds of motor proteins. This is actually the myosin family. There's always a motor domain, and that motor domain always has an ATPase binding site. And so it binds ATP and splits it. So that's why you call this the motor domain. Okay. Now, focusing on motor proteins that bind to microtubules, remember I said that if you want to walk from the negative end towards the positive end, you use kinesin. Okay. And so we're going to focus on that guy. If you want to, and we'll also look briefly at the other guy that moves from the positive end towards the minus end, that's called diamine. So those are the two players here, right? But they're both splitting ATP. They both have motor domains, and depending on what they do, you get movement of traffic in different directions along the microtubule. So you can actually see why the filament has to be polarized so that the motor protein knows which way to move the cargo. Okay. And so let's look at the gross structure of a kinesin molecule. So this is a positively directed microtubule motor protein. It is a heterotetramer, so it's got 4 pieces. It's got 2 heavy chains that are coiled together, so that's a coiled coil motif. And then it's got an ATP globular motif at the other end. Okay, so this is going to be the engine. It's going to split the ATP, and then it's got a little hinge region. We call this the neck linker. It looks like the neck of the head, right? So it's a linker, and that turns out to be really important. It's a spring-like device. And then this is the dimerization domain coiled around it. And then the thing that's going to carry the cargo is the light chains. So there's 2 light chains bound to the heavy chain at this end. And it's the light end that chooses the cargo. The light chains bind the cargo directly. Okay? All right, so we're going to walk along. So this is the end that's going to bind to the microtubule. So it has 2 functions, right? It's going to split ATP, and it's going to walk on the microtubule. So what does that walking look like? And you've already seen it because it was in that movie I showed on the first day of class. So we've come completely full circle. Okay, so let's take a look at that. And I hope this doesn't blast us out of the park here. So what you're looking at, and I'm probably going to stay pretty close to this. So this is the microtubule right here. Okay? And it's got an alpha, beta, alpha, beta, alpha, beta subunit. And this thing is the kinesin. Okay? And so this is an ATP binding domain, and this is an ATP binding domain. Note that it has 2 because it's heterodimerized, right? Okay? So these are the heavy chains, and these are the ATPase domains. We call the one out in front, we call that the leading head. Even though it's walking, we don't call it a foot. We call it a head. Okay? And so the leading head is the one out in front, and it's trying to go and bind to a beta subunit of tubulin. Okay? The lagging head is the one that's back. So when you're walking, one of your feet, and just pretend that these are heads. So you're walking on your head, right? The one out in front is the leading head. The one in the back is the lagging head. In microfilament world, or microtubule world, the movement of these two heads must be coordinated. They have to work together, okay? And so we're going to start initially with both of these heads bound to ADP, not ATP, ADP, all right? And so let's run through this, and this has some narration, I think, if I've got the sound right. Okay, no sound. Let's see if I can figure out what's wrong. It'll probably be blasted. Okay, well, guess what? I get the narrator for you. All right. I'm sure she has a much more pleasant voice than I do, but let's start over. Okay, so starting here, they're bound to ADP. So let's go forward, and I'm going to stop as we go along. So it's going forward. So you can see that both of them are bound to ADP. Okay, now there's the leading head. Okay, what happens is the leading head touches the beta tubulin. Okay, the beta tubulin acts as a adenine nucleotide exchange protein, not a GEF, but a ANEF, an adenine nucleotide exchange. What does that mean? It means that, as you can see, it's going to lose the ADP and it's going to pick up ATP. So this isn't a nucleotide exchange reaction, just like a G protein, right? Except that it's an A protein, if you want to think about it that way. All right, so the leading head touches down and exchanges, it loses ADP and it picks up ATP. Okay, now why is that important? Okay, did you see what happened there? Let's go back just a little bit. So when ATP went on, what happens is that causes the neck linker, which has this spring-like action, to fold against the microtubule. It pulls down and docks with the microtubule. That's what ATP binding does. Okay, and because you're pulling on the linker, it's going to swing the lagging foot forward, and it becomes the leading foot. Okay, so let's go back and watch that. So we get ATP exchange. Okay, then that linker is going to dock, and that's going to swing forward the next head. Okay, and it's going to bind and it's going to exchange, and so you can look at the front head all day long, but there's something going on with the back foot too. Okay, so this is now the lagging head. It was our original leading head, and so after the neck linker docked, it became the lagging head. And look what happened to it, though. Let's go back. Okay, so what's going to happen is this one is swung forward. So this one used to be back here. It swings forward. This is now the lagging head. The linker's docked. Look what happens next. It splits ATP. Okay, and that's critical because splitting ATP allows the lagging head to lift up. That's what unbinds the lagging head from the microtubule. And why is that important? Because it's about to be swung forward to become the leading head. So the rule is if you're bound to ADP, you're not bound to the microtubule. Okay, you release, you quickly release from the microtubule, particularly if you're the lagging head. Right? Okay, so let's just let it go through the cycle. ATP hydrolysis, it makes that little gas come out here. No, it doesn't happen. Okay. Okay, and if it's not on your DVD, it's on the website. It should be within this lecture. Okay, questions about that before we go on? Yeah?
Does the linker binding ever go backwards?
No.
Because of the polarization?
Yeah, it does not go backwards. Now, what you have to do is there is an isoform where the position of the linker has changed. It's been mutated or through evolution, and it walks the other direction. So there's a few kinesins that can walk to the minus end, but they're rare. Okay, and it's all about where you position the linker determines which direction you move. Okay, and we also call this this has a high degree of processivity. In other words, it walks forward very carefully for a long period of time. So it's really good to use something that's going to move a long direction, like a neuron carrying, like a kinesin carrying a secretory vesicle full of neurotransmitter. You don't want it to fall off, right? And remember, there can be dozens of these things all walking on the same microtubule. I actually have a video of two vesicles that actually cross each other. They pass each other on the same microtubule. So there's lots of binding sites, and there can be multiple motors moving along this. Okay? Cool machine. So we'll put that away. That. Okay, where does that bring us to? Okay, so that's the picture, we did that. Okay, now, if you want to walk the other direction, you got to pick a different motor, unless you have that really weird version. But as far as we're concerned, all kinesins move to the positive end. That's a good generalization. All right, now let's talk about dynin. And we're not going to go through the same deal because it is a really complicated machine. So I want to point out a couple of differences. The key thing is that dynin moves cargoes to the negative end, okay? That's the key thing. Again, it's going to have ATP binding domains that contain the motor domains that split ATP, okay? We're not going to go through the binding cycle because it's much more complicated. But the key thing is, the other difference is that dynin doesn't actually bind to the cargo. It binds to this really complicated thing called a dynactin complex, and that's what actually binds to the cargo. So it's a much more complicated device, but essentially it still splits ATP, but it moves in the different direction to the minus end. Okay, so you'd use this if you wanted to carry something from the plasma membrane towards the nucleus, okay? Or at least anywhere towards the negative end. Okay, and that's all I really want to say about that. So, any questions? Now, let's see. Now, which way? If I could go back quickly to myosin, which way does it walk? Positive or negative? Myosin 2 always walks to the positive end. Always walks to the positive. Okay, so it's a plus end directed microfilament motor, whereas this, how would you characterize this? This is a negative end directed microtubule motor. So the highway that you're walking on matters, and the direction in which you're walking also matters. Now, I show this slide because we had said very much at all about microfilaments, except for that real brief characterization. Remember, this is intermediate filaments. Okay, so given the name, it's intermediate in diameter. What is its construction? Rope-like. It doesn't bind nucleotides. It's not polarized. There's no plus or minus. What's its function? Pretty much structural integrity. There's no movement along it, there's no cargoes moving along. It's there to tie things together, okay? What gives it its diversity? The different isoforms of the intermediate filaments is what gives it its diversity, okay? So if it's a certain type up here, if it's made out of lamins, those are intermediate filaments, that's what makes your nuclear lamina, okay? And you can do all kinds of different things with intermediate filaments, but it's dependent on the isoform of the filament itself, not the binding partners. That's what's different. It doesn't have very many binding partners. It's the filament that it's very different in different tissues. That's the key point. Not to memorize this. It's the key point is diversity, functional diversity comes from the different isoforms. And there's very few isoforms of actin and tubulin, but there's lots of isoforms of intermediate filaments. Okay. So, structural integrity. Okay, any questions about the cytoskeleton before we move forward? Okay. So now let's turn to the cell cycle, which is going to be our last topic. Okay, so what is the cell cycle? It's a ordered series of events by which a cell grows, duplicates its DNA, and divides. So if you're in the cell cycle, you're in there for a reason, and the reason is to divide. So if you're a stem cell, you will be in the cell cycle, and you won't leave it, right? Other cells can exit the cell cycle, and you can come back, but there's some rules about that. But basically, you have two different decisions to make if you're a cell. It's really that basic. If you stay in the cell cycle, the goal is to divide. Okay? If you leave the cell cycle, there's two things you can do. Okay? What you can do is you can leave the cell cycle and kind of repair any damages, or maybe if you're not big enough, you can get a little bit bigger. But basically, that's one of the things you can do if you leave the cell cycle. When you leave the cell cycle, where do you go? That state is called G sub 0. Okay? And we'll place it in this little order here in just a moment. The other thing that you can decide to do is if you differentiate into a really highly specialized cell, you're not going backwards. So neurons don't form neurons and then somehow collapse back into a little ball and then divide. That doesn't happen. Okay? There's very little dedifferentiation that can occur and lead back into the cell cycle. It's rare. Okay? So basically, you leave the cell cycle to repair things, get bigger, or just to wait, or you do that so you can differentiate into a specific cell function. That's the whole science of developmental biology, is what happens in this step right here. Okay? So you have to exit the cell cycle to differentiate. All right, what does it look like? All right, so the cell cycle consists of 4 different parts. Okay? It's got G1, which has the name gap 1. It's the gap between 2 basic events. One is mitosis, which is very recognizable. So when cell biologists a long time ago were trying to figure out what the cell was doing when it divided, and then it became 2 daughter cells, and then it kind of grew and it divided, there were 2 things that were very obvious. One thing was the cell undergoing division, and we call that M phase. Okay? And notice that M phase, as I mentioned, has both nuclear division steps and it has cytoplasmic division steps. Okay? And then there was this big gap, and that's when it spent a lot of its time just getting bigger and accumulating nutrients. Okay? This is also the period at which the cell can exit the cell cycle. So G0 is when you exit G1, that's where you go. You go to G0. You can't go to G0 just anywhere. You can't decide to leave an S phase, which is synthesis phase. That doesn't make a lot of sense. Why would you synthesize, duplicate your DNA, that's what's happening in S phase, and then leave the cell cycle and you'd have too much DNA, right? Because if you double your DNA, you need to divide it to put it into 2 daughter cells. If you exit the cell cycle here, that's wrong, okay? So if you exit in a controlled manner, then you leave G1 into G0. Can you come back in? Yes, you can actually, if you don't differentiate, you can wait there, get bigger, and then maybe you get a growth factor put on you by an investigator, by a pipette, squirt some epidermal growth factor on top of you, then you'll go back into G1 and continue dividing, right? Okay, so you go from G1, S phase, that's where you duplicate your DNA, and duplicate the centrioles. Then you go into gap 2, this is where you get a little bit bigger, and several different proteins get produced, and then you go into mitosis. All right, so these are the 2 shortest phases, these are the 2 longest phases. This is actually really short. It's in a typical cell, it's in the cell cycle. This would be, if this was a 24-hour clock, which it rarely is, but if this is 24 hours, this is about 30 minutes. This happens very fast. Okay? Now, if we look at the outline, if you are in the cell cycle, you have to proceed through different checkpoints. You don't just go flying through this thing. There are different checkpoints, okay? And those checkpoints, there's actually probably, you can make an argument for more than 3 checkpoints, but these are the main ones that we're going to focus on, okay? The first checkpoint is called start. That's a good name for it. It's also, mammalian cell biologists tend to call this restriction. It has 2 names. It was actually named by the yeast people that named this cell cycle, and that they call it start. Okay? Where is start? It's at the end of G1 and right before you get into S phase. So this is a big decision. Am I going to go into S phase and use all my energy to duplicate my DNA? If you're going to do that, make that decision, you better be really committed to going through that. Okay? So you got to pass this checkpoint. The second checkpoint is the gateway into mitosis. We call this, it has various names. Your book calls it G2M checkpoint, which is kind of clumsy. I call it the mitotic entry checkpoint. ME, mitotic entry, okay? That's a big commitment, isn't it? Because your chromosomes better all be duplicated, and they better be ready to be pulled apart, right? So that's a big checkpoint. The third critical checkpoint is actually partway through mitosis, and that's called mitotic exit. And that checkpoint is at the interface between metaphase and anaphase. Do you remember what metaphase is? That's when all the chromosomes line up at the metaphase plane. That's the last chance you have before you segregate the chromosomes and they're pulled apart. That's called anaphase, right? So it's the transition between those two. That's absolutely essential, that everything's lined up. What if some of your chromosomes are not attached to microtubules? What's going to happen when you pull apart? You'll have misjunction aneuploidy. So one cell will get too many chromosomes, the other cell will get too little. It will probably die or become cancerous or whatever. It's a screwed up mess. Okay, so this is a really critical step. We're going to focus our attention on these two because of the lack of time. And so the question we're asking, what does it take to get through a checkpoint? Generally, it takes 2 things. You have to have what's called the cell cycle engine activated. Let's take this one. Well, since we're going to focus on the mitotic entry, let's use that as our sole example. All right, so to get across this checkpoint, the flow of the cell cycle is in one direction. It cannot go backwards, and there's reasons for that. The cell cycle is unidirectional. So if this is a river, it flows clockwise, okay? And the reason I'm bringing that up is the 2 things you need to get past this checkpoint is you have to activate what are called the cyclin-dependent kinases. These are the cell cycle engines. So it's a protein kinase, and it's a protein that you know of what its function is. You take ATP, you take a phosphate, you stick it on a protein. That's a kinase. But they're very, very special kinases, and they work by having a regulatory subunit called cyclin, and then the kinase is a separate subunit. So you have to activate that kinase. That's number 1. And that's called the cell cycle engine. The second thing is you have to have completed all the events downstream from the previous checkpoint. So if you're approaching this checkpoint, you're going right here, you have to look behind you and say, have I completed all the things that happened since this last checkpoint? Okay? So downstream from this checkpoint, this was the previous checkpoint, these events would be downstream, these events would be upstream, right? So if you're walking along a river, if you're going downstream, you walk this way. If you go upstream, you walk this way. So the rule is, to get past this checkpoint, you have to have completed all of the processes downstream from the previous checkpoint. So what are those? You go to the previous checkpoint, and what happens during all this stuff? Well, number 1, did you duplicate your DNA? Number 2, did you duplicate your centrioles? Why do you need to duplicate your centrioles? What's going to happen in mitosis? you're going to have two poles, so you better have two microtubule organizing centers for the spindle fiber to come out of, right? You better not have any DNA damage because you're about to go and form a new cell. So is my DNA repaired? Is it duplicated? Do I have centrioles? And then what happens here, the downstream event, here's the previous checkpoint. So downstream from that is you better get big enough. So during gap 2, you need to make all the proteins you need because you can't do that during mitosis. Why can't you synthesize proteins during mitosis? Pardon me?
You're going against the chromosomes.
So you can't turn on transcription, right? And so all things are all messed up. Okay, so you don't want to. So this is your last chance to make any proteins and get really muscular. And then, in fact, there is a size restriction on normal cells to go into mitosis. If you're not big enough, you're too weeny, you're out of here. Okay, you're not going through. If you're a cancer cell, you don't give a poop. Okay. You're tiny. That's why cancer cells are so tiny. They don't care if they're small. They don't care. That's why they look so. If you look at a histological slide, you'll see normal cells. They'll be a normal size, and you'll have all these little messed up guys that are tiny. That's because they're going through this so fast that they never build mass. That's the hallmark of tumor cells. Okay. So normal cells are not like that. They have to complete their size equivalent before they go into mitosis. Yes?
Do they get destroyed if they don't get big enough?
Yeah, so what happens to you? If you're not if you're in G1, you can exit into G0. But if you're over here, there's only two possibilities. You can arrest in that phase and repair or get bigger, or you have to kill yourself. Those are your two choices. You have to undergo apoptosis. Okay, and that's listed up there. Apoptosis, and that word is not ptosis. I'm not spitting on people. It's ptosis. It's apoptosis, and you'll never hear that again. And everybody calls it, because they're lazy, apoptosis, and that's just wrong. But that's okay. All right. So here's the machine. This is a cyclin-dependent kinase. This is the enzymatic part that's going to take ATP, pull off a phosphate, and stick it on to some substrate that fits in here. And this is the regulatory subunit. So this is a heterodimer, right? And the one we're going to look at is the one that's necessary to go right through here, the mitotic entry phase. Now, are there several different kinds? Absolutely. So let's understand that there are different cell cycle engines for the different checkpoints. But the one we're going to focus on is the mitotic CDK, cyclin-dependent kinase. And this is the only one that you actually have to know the precise names of the two proteins. It is, the mitotic CDK is composed of CDK1, that's easy to remember, and cyclin B. So that's the only time I'm going to ask you for the specific components. But understand that we know what the other components are. This is our poster child, mitotic CDK. Okay? And that's the one to get into mitosis. All right? So what's going to happen is throughout the cell cycle, from G1 all the way to, here's what we want to try to get through. There's always plenty of mitotic CDK. Right? The CDK, so that's CDK1. CDK1 is not the problem. The problem is it has to have its binding partner. The thing that changes in concentration during the cell cycle is this little green guy. That's cyclin B. So it's cyclin B is the determining factor of whether the engine works or not. I'm actually going to use a car analogy to bring that home. So the reason it's called cyclin is that some smart fellow realized that the protein cycles in its concentration throughout the cell cycle. They went, aha, that's probably what's telling the cell to go through the checkpoints. Okay? So that's exactly what you have to have this thing fully assembled, and so you have to have the M cyclin, which doesn't get high enough until G2. So that's the reason you're waiting for G2, you're waiting for the cyclin B to get high enough in concentration to push you through the checkpoint. Okay? Now, if it only was that simple. What's the other thing you have to, what's the other thing? You have to have the engine assembled, and don't forget the second thing. You have to have completed the downstream events from the previous checkpoint. So you have to go back to the previous checkpoint and ask, did I finish making my DNA? Is it repaired? Do I have centrioles? And am I big enough? Those are the downstream events from the previous checkpoint. Okay, so if all of that is in order, what's going to happen is the following. Okay, so think of, this is CDK1, all right? That is a serine threonine kinase. Remember, it's a kinase, you always have to know what kind of kinase it is. So it phosphorylates serine and threonine, so it's kind of a Joe Blow-like kinase, but it really isn't. Okay, so CDK1, it is completely inactive without its binding partner. So you can think of it as a car, and we're going to use a manual transmission. Can everybody drive a manual? Oh, not. Okay, but enough of you know. So CDK, that is a car engine that's in neutral. It's sitting there idling, okay? Your foot's on the brake, not going anywhere. But it's prepared to go someplace. When you when cyclin B becomes synthesized and reaches a certain concentration, the two of them stick together. Now, at that point, that's like putting your car into first gear. You're ready to go forward at a pretty slow rate, right? So there's a little bit of activity that's caused by cyclin B binding to CDK. The reason that activity is there at all is because what you've done by these two binding together is you've taken what's called the T loop and you've bent it downwards, okay? What happens is when this is all by itself, this little red T loop is blocking access to the substrate binding site. It's blocking, okay? And so if you take your cyclin B and push the lip of that downwards, you've now created a little space that a substrate molecule can get in. So you're in first gear. So think of the cyclin B as the stick shift putting you into first gear. You're not going very far very fast, right? Okay? Now, what actually happens next is you actually put your foot on the clutch, but let's just ignore that. Let's just pretend this is just a manual transmission or no, automatic transmission. You don't have a clutch. Okay, what's going to happen is a second kinase is going to come in. It's called, strangely enough, a CDK activating kinase, or CAC. Okay, and CAC is going to phosphorylate the T loop on threonine 160. What that does is it just drops the loop down and allows full access to the substrate. The rate of catalysis goes up 300-fold. Okay? So what is CDK, the CAC? What's the analogy? Putting your foot on the gas. Okay? Now, what really happens, though, is you're driving a manual transmission. Okay? Now, what happens is this is idling by itself, no kinase activity at all. Okay? And what happens next is the cyclin comes in, you put it into first gear, so it's ready to go a little bit to start, but what happens is, the next thing that happens is an inhibitory kinase sneaks in, okay? And this is called, it's got a funny name, it's called WEE1, okay? WEE1. And the reason it's called WEE1 is if this thing is mutated, you get wee little cells, because it disrupts the cell cycle controls. Okay, WEE1 is a kinase, okay? And it is not just any kinase, it is a tyrosine kinase. And it puts a tyrosine onto a phosphate onto tyrosine 15, okay? And that is an inhibitory phosphate. So what WEE1 is doing is it's putting its foot on the clutch. Can you go anywhere if your foot's on the clutch? Doesn't matter what gear you're in, it's going to go nowhere, okay? And what happens next is the activating, the CAC comes in. So the actual order of this is CDK binds the M cyclin. It starts to go forward a little bit. WEE1 stops it by putting in the clutch, and then CAC activates it, So what are you doing? You're revving the engine. That's your accelerator. The CAC is the stimulatory activating phosphate on tyrosine or threonine 160. So you're standing there with your foot on the clutch, pushing the accelerator. What does your engine do? You know, the RPMs go through the roof. They actually go 300 times faster than they were in first gear. Okay, so what do you have to do to just speed off? You have to pop the clutch, right? And a lot of you do that, you get caught by the policeman, don't you? It takes off. So if you pop the clutch, you're gonna shoot forward, and the cell is gonna go right into mitosis. Okay? What pops the clutch? What pops the clutch is it on here? Oh, there it is, right up here. This is your foot being pulled off the clutch. It's called CDC25. It is a phosphatase. Now, have we seen a phosphatase activating proteins before? We had, what about calcineurin? You know, calcineurin would dephosphorylate that transcription factor. Well, this is what's happening here. You don't take off both phosphates. Which phosphate do you take off? The inhibitory one. So CDK25 is a phosphatase it's a tyrosine phosphatase only. And so it will clip off that, so your foot comes off the clutch. Your accelerator has already put one right there, so you're already got your foot on the gas. So this thing is going to fly forward. Okay. So this is what pops the clutch. It is a phosphatase, tyrosine phosphatase. Now, what activates that? Okay, it is activated by phosphorylation. So what's going to happen is if you activate this CDK, which is a kinase, it's going to phosphorylate more CDC25, which is going to pop the clutch on more of these, and so you get positive feedback because this activates that. Okay, and the other thing is, so CDK phosphorylates a lot of stuff. The mitotic CDK, it phosphorylates CDT25, but it also phosphorylates WEE1 and turns it off. So there's two reasons this goes through the roof. Number one, CDK is activating its own activator. And number two, it's deactivating its own inhibitor. Those are the two reasons that the activity, not the concentration, the activity of kinase, CDK kinase just goes like this. And that's, throughout biology, when you see a spike like that, it's positive feedback. Okay. So this is a little complicated, and I don't know if the car analogy works for you, but if you understand the order of when things happen, this will make sense. Are there any questions about what's going on here? It's a neat machine. Okay. All right, so this is really the key to proceeding into mitosis. What's happening? What's going to happen next in mitosis? Talk about all this stuff. Put it all away. Okay, so you know it must. CDK is going to phosphorylate something that allows all of these early events, prophase, prometaphase, metaphase, to occur. What's going on here? Chromosomes are condensing. The spindle fibers are forming. The poles move apart. The spindle fibers attach to the chromosomes. They line up at metaphase plate. Those are the events that CDK has to bring about. How does it do all those important things? It's just one kind of kinase. Well, it has lots of targets, okay? Number one, if you're going to take a normal cell that has polymerized microtubules and create two different spindle fibers, you're going to have to deconstruct the whole thing. So what you have to do is you have to destabilize all of the microtubules and rebuild them into spindle fibers. How can you do that? You phosphorylate the MAPs that are stabilizing. So one of the targets of all CDKs is MAP. It's actually MAP4, I guess. It tends to be in all cells. So you phosphorylate this, it's going to lift off. The tubulin will come apart, and it can be reformed into spindle fibers. So you actually destabilize microtubules to make the spindle fibers. So that's real obvious. Okay? This is obvious too. You phosphorylate histone 1. What does that enable you to do? This goes back to the first quiz 1 material. If you phosphorylate histone 1, it activates it and it causes chromosomal tight packing. You form the 30 nanometer solenoid instead of having loosely flopping DNA that's fully activated. You want to pack the DNA. So that's the idea. To do that, one good way is to phosphorylate histone H1. Okay? Now, this is another protein that we talked about early on. Condensins, what do? They are structural proteins that form the backbone of each chromosome. And so what you want to do is turn them on and have them supercoil or pull together all the DNA into a tight coil. And to do that, you phosphorylate them. Okay? So it's as simple as that. So phosphorylating the condensins allows them to pack DNA even tighter into a mitotic chromosome. Okay? The next thing we need, after you pack the chromosomes, you've reformulated the spindle fibers. The spindle fibers will do you no good if you can't get into the nucleus, right? So what are you going to do? You're going to break down the nuclear envelope. And the trick to breaking down the nuclear envelope is to simply phosphorylate the lamins, the intermediate filaments that make up the nuclear lamin. If you can break this apart, the membrane will dissociate and the chromosomes are now available to attach to microtubules, the spindle fiber. Okay? So phosphorylation breaks down the nuclear envelope. Okay? And the last thing you want to happen at this point is for the cell to divide, or is to undergo cytokinesis too early, right? That will screw up the distribution of chromosomes. So you want to inhibit cytokinesis. How can you inhibit cytokinesis until later? You can phosphorylate myosin 2. You prevent the little purse strings from pulling by phosphorylating myosin 2, the motor protein that makes contraction possible. So this is an inhibitory phosphorylation of myosin 2, the motor protein. Okay? So you're going to prevent this from happening, right? Until the right moment. Okay? And this is myosin 2. What you phosphorylate are these little light chains up here. Those are a target of CDK1 phosphorylation. Okay? So now you have this, right? And then you should have spindle fibers attached to it there. And you're ready to pull, right? Okay? So that's the next job. So if everybody's attached and we pull it apart, then we should be able to go forward. This is the next checkpoint. This is the third checkpoint. So we had start, we had mitotic entry, which we just covered in detail. Now we're going to cover the last one, and that's the last topic we have to talk about. Okay, so this is mitotic exit. How do you know when it's time to pull them apart? Okay, so this is the kinetochore. These are all attached there. This is a nice spindle fiber that's attached to, which spindle fiber is just another name for microtubule. Okay, now what's holding the two sister chromatids together is this protein called cohesins. Okay, so it's linking. It's like a little hand charm bracelet, right, around the chromosomal, the two sister chromatids. So the key is to break this in two, unlatch the hinge. How can you do that? Well, this can be attacked by a protease, which is the aptly named protein separase. So separase would like to cut this right there, and the two proteins can come apart, right? But what activates that? So it's the control over separase is what controls when the two come apart, right? So if we can answer that, we can pull them apart, okay? What happens is we have a protein that's called anaphase promoting complex. And so this is one of the key things that controls what's going to pull the chromosomes apart. And so what happens is CDK phosphorylates it. So that turns on APC. All right? Now, APC cannot do its job unless it has a guidance molecule. This is a targeting molecule that's going to say, I want you to go target separase, and I want you to activate it. Okay? So the phosphorylation of APC allows the targeting device, which is called CDC20, to latch onto it. So now this is a weaponized protease that has a targeting device. Okay? Where does it target it to? It targets it to separase. Okay? Now, what does it do? You don't want to chew up separase. You want to chew up this protein here, which is holding it inactive. That inactivator is called securin. And so it secures the cell cycle until you're ready to break the chromosomes apart. So all you have to do is, this protein here is going to attach ubiquitin to securin. What's that going to do to securin? If you polyubiquinate a protein, where does it go? To the proteasome. So that's what happens. This is a ubiquitin ligase. That's the activity of APC. It's targeted to this structure, and it attaches ubiquitin to the securin. That is destroyed, leaving separase fully active. What is the activity of separase? It's a protease. What does it chew on? This necklace. So the separase will nick that, and then the two sister chromatids can come apart. Because you're already pulling on them, right? The spindle fibers are already tugging on them. Okay? Now, a related problem is when we do that, what we have to do is also we need to destroy the cell cycle engine so we can't go backwards. Okay? And fortunately for us, APC also ubiquitinates in the CDK. The complex. What does it actually destroy? It actually destroys cyclin B. Is that as good as destroying the whole complex? Yes. It doesn't destroy the enzyme, it destroys cyclin B. So this thing will also ubiquitinate cyclin B, destroy it. Okay? Now, you have, the only difference when you do that is you use a different targeting device. Instead of that CDC20, it is CDH1. It has a different headgear, okay? And the CDH1 targets it to the cyclin B and puts ubiquitin on that so that it's taken to the protease and destroyed. Okay. The last thing we really have to answer is this one up here. And how does the cell know that every chromosome is attached to spindle fiber? How does it know? Okay. What happens is if there is no spindle fiber attached to the sister chromatids, what happens is these proteins that have funny names are called MAD and BUB. MAD and BUB will attach to the kinetochore if there's no spindle fiber. Okay? And what happens is if you block the kinetochore, it will actually bind to CDC20. So MAD and BUB, when they're attached to the kinetochore with no spindle fiber, they soak up all the CDC20 in the cell. Why do you need CDC20? It's the targeting device for APC. So this can't go forward until all of the MAD and BUB has been displaced from the kinetochore. Guess what displaces MAD and BUB? The spindle fiber. So the spindle fiber comes in here. If it attaches to the chromosome, MAD and BUB will leave and they'll let go of CDC20. CDC20 now is the targeting device for APC and targets it. Where does it target it to? To separase. Actually, the part of it is securin that it's going to destroy. So that releases CDC20 to do its job. So that's how you actually, it can detect whether there is a spindle fiber attached or not. So now you pull them apart, and what happens is if you destroy the mitotic CDK, what happens is all the phosphatases in the cell now take over, and they remove phosphate groups from all of those things that we phosphorylate. So we dephosphorylate the MAPs, we dephosphorylate the histones, we dephosphorylate myosin 2, we dephosphorylate the lamins. So everything comes back together because of dephosphorylation. And that's what allows you to go through mitosis and into cytokinesis. And that's where we will draw the line. So let me point that out. So after this slide, that's the end of this material. I put the other material in there just for your own edification, but this little dividing cell will be your image, that's the end of the final exam material. Those of you that are interested in how growth factors drive a cell into the cell cycle can look at the rest of it. Okay. And the line here on the outline, in case you brought it with you, is right here. We will not cover mammalian cell cycle controls. That's right here is the end of the final exam material. I will repost this PowerPoint and say this is the end of the material, so you'll have it. Thank you very much. We'll see you on Thursday, bright and early.